Consider a powerful tool that could change life regulations. It would help medicine, farming, and even our understanding of how evolution works in a big way. That's what CRISPR, a cutting-edge tool for changing genes that makes science fiction real, says. CRISPR can be used to fix genetic diseases, make foods resistant to diseases, and do many other things. On the other hand, enormous power comes with enormous duty. In this video, we'll go over CRISPR technology in more depth. We'll discuss its origins, uses, and issues. Let's get to know CRISPR technology closely. CRISPR is a group of DNA sequences found in prokaryotic species that are used to protect against viruses. It's in the genomes of 90% of archaea and 50% of bacteria. CRISPR-Cas9 technology edits genes with CRISPR sequences. It can be used in biology, science, and treating diseases. 3 different places around the world separately found clustered DNA repeats, which became known as CRISPR. Yoshizumi Yoshino and his colleagues first wrote about CRISPR in 1987. They cloned part of a CRISPR sequence along with the IAP gene from the genome of Escherichia coli by mistake. In 1993, 2 papers were written by Mycobacterium tuberculosis experts about a group of interrupted direct repeats, DR, in that bacterium. At the University of Alicante, Francisco Mojica looked into how repeats work in the archaeal species Halopharax and Haloarcula. Rodolphe Barangu, a yogurt expert, found in 2005 that adding more CRISPR spacer sequences to Streptococcus thermophilus makes it more resistant to phages. Scientists think that the CRISPR-associated systems, which are made up of Cas genes, helicase motifs, and nuclease motifs, play a part in how bacteria adapt to their environment. Scientists have used the CRISPR-Cas9 system, a less complex version of the CRISPR system from Streptococcus pyogenes, to change the genomes of many different types of animals, including human cells. It has been shown that the changed method can edit genes effectively in human tripronuclear zygotes, which lets DNA be cut more than once. Cas12a is a nuclease from the Francisella novicida bacteria that is better than Cas9 in a number of ways. It can make a staggered cut in double-stranded DNA, has a T-rich PAM, and can be targeted with CRISPR RNA-CARNA. In the DNA, Cas13a cuts 18 to 23 base pairs downstream from the PAM site. It does this by following RNA instructions. This lets DNA be cut more than once. These characteristics have made many different diagnostic technologies possible. CRISPR arrays are made up of a leader sequence that is high in ATs and short repeats that are divided by unique spacers. They can have anywhere from 28 to 37 base pairs. There are 35 groups of Cas genes, and 11 of them make up the Cas core. CRISPR-Cas systems are split into 2 main groups and 33 smaller groups. Many living things have more than one system. Bacteria and archaea naturally have a process called CRISPR-Cas immunity that breaks down alien nucleic acids to stop bacteriophages from infecting, conjugating, and changing naturally. Cas1 and Cas2 proteins help with spacer acquisition, which is an important part of the immune response. In E. coli, Cas1 and Cas2 work together to bind double-stranded pieces of DNA that are trying to get in and add new gaps to CRISPR arrays. For type 1 and type 2 systems, protospacer adjacent motifs, PAM, are important, but not for type 3 systems. Sulfolobus sulfataricus. Multiple CRISPR spacers can be found on a phage. This is called priming. This occurs when spacers aim for the phage, even if they don't match the protospacer. Once the alien DNA has been primed, the acquisition machinery slides along it to find a new protospacer. There is a CRISPR pattern that turns into cRNA, which is then cut up by Ca's proteins. In type 1 systems, interference depends on the cRNA and the protospacer having the right base pairs. In type 2 systems, it depends on Cas9, a single protein that can do many things. Type 3 systems use a different Ca's protein to target DNA as well as RNA. It is thought that the CRISPR-Cas system, which can protect bacteria against some phages, came about from 2 earlier systems. The Cas1 and Cas2 genes in the adapter and effector modules are very similar to each other. The class 1 effector Cas genes have evolved from the original effector module. The spacer regions of CRISPR-Cas systems come straight from foreign mobile genetic elements. This makes it difficult to track how they have changed over time. CRISPR-Cas can make cells immune to some phages, which stops the spread of disease. CRISPR sequences show how the host and virus genomes change together over time, with different rates of spacer acquisition. Researchers have found that phages can infect and multiply in hosts with just one targeting spacer, and that situations with a lot of phages can still have sensitive hosts. CRISPRs are a type of DNA recombination protein. Their repeating spacers and straight repeats make them simple to spot in DNA sequences. However, it is challenging to analyze CRISPRs in metagenomic data because they are repeated. PCR, degenerate repeat-specific primers, and shotgun metagenomic data are some other methods that can be used. Phage-inducible chromosomal islands, PC, are a way for bacteria to protect themselves from phage attack. PCAs stop phage replication in several ways, such as by attaching to phage terse, shifting capsid morphogenetic protein, and going after LTRC. ICP1 phages have a CRISPR-Cas system that targets PC-like elements. This lets the phage and host evolve together. CRISPR gene editing is a technique that has been used in many fields such as farming and food to create probiotic cultures, protect industrial cultures from getting infections, and improve crop yield, drought tolerance, and nutritional value. In addition, it has become an important tool for scientific study because it makes it possible to multiply and knock out gene products to find genes that could be used to make new medicines or help us understand how complicated genomes are. About 1,000 research articles had been written about CRISPR by the end of 2014. In human cell lines and cells, CRISPR has been used to turn off genes, study Candida albicans, change yeasts used to make biofuels, and change the genes of crop types. In 2019, CRISPR was used to treat a person with sickle cell disease. In February 2020, 60 to 80% of integrated viral DNA was cleared from mice as part of progress in HIV treatments. In March 2020, a CRISPR-modified virus was put into the eye of a person with Leber congenital amaurosis to help them see again. CRISPR gene editing could be used in the future to make new species or bring dead species back to life by using closely related species. CRISPR-based reevaluations of claims about gene-disease relationships have found some strange but possibly important things. Researchers used CRISPR to change the genes of high PSCs in July 2021 to look into the role of MBNL proteins linked to DM1. CRISPR has been shown to help with a number of diseases, including those affecting the nervous system, the circulatory system, stem cells, blood disorders, and muscle degeneration. Hemoglobinopathies, hemophilia, neurological disorders, blindness, and cardiovascular conditions are some of the uses. Hemoglobinopathies occur due to structural changes in hemoglobin or the substitution of different amino acids in globin chains. This can cause red blood cells to stop working, blood vessels to get clogged, growth problems, and vision issues. CRISPR-Cas9 makes it possible to move multipotent cells into mouse models so that gene therapy rates and gene correction can be studied. This extends the half-life of red blood cells. CRISPR has also been used to stop mutations that make functions better and fix mutations that make functioning worse in neurological diseases. CRISPR works by grabbing the gene that codes for a retinal protein and changing the genome to make vision better. CRISPR is a technology that was created in 2013 and has become an important way to change the genes of plants. To get more serial, the balance of cytokinin, an enzyme that codes for cytokinin oxidase dehydrogenase, CKX, has been changed. This increases the production of carbohydrates similar to wheat, but the quality of these carbohydrates decreases due to the presence of more gluten proteins. Bacterial attacks on rice can turn on the transcription of genes, creating lines of resistance. This is another way that CRISPR helps plants deal with biotic stress. CRISPR has been used in gene therapy to replace broken genes with DNA from outside the cell and change sequences that have been changed. Base editing can be done with two different types, the cytidine base editor C and the adenine base editor ABE. The ABE can only fix up to 4-point mutations and edit point mutations. To get around these problems, the CRISPR system has added a new method called Cas9 fusion to change genomes at a higher level. CRISPR can also change genes by turning them on or off, depending on the genes that are being studied. It is possible to turn genes on or off using a nuclease called dCas9, endonuclease. But when it comes to clinical applications, researchers face problems such as moral issues and getting to the target spot. The CRISPR system moves from bacteria to host cells, which makes the immune system attack the bacteria. This leads to issues such as cell death and damage. Using CRISPR-associated nucleases as molecular detecting tools for molecular testing could be useful. For example, they could be used to get rid of unwanted nucleotide sequences in next-generation sequencing libraries and test nucleic acids directly for diseases. The goal of CRISPR diversity research is to find out about phylogeny and diversity in bacteria, like xanthomonads. Molecules other than nucleic acids can be found by connecting CRISPR-based tests to other enzyme-based processes. Sherlock-based profiling of in vitro transcription, SPRINT, is one example. It can find many drugs quickly or with portable point-of-care devices. The CRISPR-Cas systems are also being looked at to find and stop SARS-CoV-2, the virus that causes COVID-19. Two complete diagnostic tests, the AIOD CRISPR test and the Sherlock test, have been found for SARS-CoV-2. They both provide a strong and accurate way to see viral nucleic acid. CRISPR is a tool that can precisely change genes. It could completely change fields like medicine and farming. A big step forward in genetic engineering is that it can now fix genetic diseases, make crops more resistant to damage, and even bring extinct species back to life. However, with this power comes the duty to figure out what the moral and social effects are. Knowing CRISPR's history, how it works, and its potential uses helps us respect its progress while also acknowledging that it must be carefully regulated and its effects on society must be considered. As we learn more about and work on CRISPR technology, it's important to keep a balance between new ideas and doing the right thing.